Hi there, my name's Don. Um, I'm a biology tutor, but I also do a bit of tuition in each of these other subjects here, GCSE physics and the GCSE biology, but essentially I'm A2 biology. Today I'm going to do a short video on respiration. Respiration is an A2 biology topic, module 5. I'm going to do a video on most of the topic. I'll try to do as much of the topic as possible, but it's quite a long topic and it's quite a difficult topic. It's a topic that's asked about frequently by students who have difficulty with it. Before I start, my normal tuition procedure or process is to supply students with a set of notes, a set of my own notes, which are carefully crafted and designed to cover the essential parts of the topic. And they're very good notes, and they're sought after by students. So you'll need, if you want to follow this video, essentially you'll need a copy of the notes, a copy of my notes, and I constantly refer to the notes, to various pages and so on, essentially so that I don't miss any of the points that I want to make on this particular topic. So you'll need a copy of the notes in front of you. There's also a set of past paper questions. These questions are hand-picked to cover the most salient parts of the topic and to cover questions that are most likely to be asked in current exam questions. So a set of notes, a set of exam questions, there's a set of answers to go with the exam questions. There's also other supplementary materials. There's a set of shorter questions, something in the region of 50 shorter questions. They're also quite difficult. These questions are, whilst I call them short questions, they're actually very searching, and I've designed them to cover questions that are sometimes out of the box, let's say, or outside the box, I should say, and that they're not directly on the specification, but they're sort of cover questions like suggest questions that you get frequently at A level, and you have to come up with an intelligent answer. As I say, to follow this video, essentially you need these items in front of you, because I constantly refer to parts of the notes and parts of the questions, and we'll go through some of the questions as well, so you need these in front of you. If you need a copy of the question booklet, of the notes and the question booklet and all the materials, just get in touch with me there on my mobile number, and just text me, and I'd send you or get you a copy, a full package of materials. I'll either post it to you or deliver it to you if you live close enough to Marfelt here. Okay, so this is, I'm going to run through the topic respiration at A2, and I'm going to go fairly fast. I need to go quite fast because it's a long topic and I want to try and cover as much of it as possible. I won't be doing any drawings on the board or anything like that, because simply it just takes too long and slows the whole process down. Any drawings or any diagrams or anything else that you need will be inside the note booklet. All the things that I'm about to say about respiration are contained in the note booklet. In other words, it's all in here, all the information you need. So you need a copy of the notes in front of you, and to be able to work through the notes. If you have a copy of the notes and a copy of the question booklet, then you can go through all the questions yourself and you have the answers. Or if you want to, you can get in touch with me, and then I'll do a Skype session with you where I go through the answers or I go through the notes or whatever. Get in touch if you want a package of materials that will help you to go through this topic. Anyway, let's start respiration. Normally for respiration, this is the last page in the notes, which is like a summary of the respiration process. Now I could put that up on the board here, but it wouldn't come up properly. You need it in front of you, and it's in the note package here in the materials, and along with the rest of the materials. So I'll make reference to it as I go along. So essentially, Respiration, a process that you came across at GCSE, whereby cells release energy, produce energy. And at GCSE, you might remember that respiration could be either aerobic or anaerobic. And aerobic respiration, you might remember, was a kind of respiration which only happened inside a cell if the cell had oxygen. Anaerobic respiration was another kind of respiration that took place when cells didn't have sufficient oxygen. And you might remember that at GCSE you'd be asked to make comparisons between the two. And so the comparisons were basically aerobic respiration, oxygen present, and amount of energy released, large amounts of energy released. And then for anaerobic respiration, no oxygen available and small amounts of energy released. Well, at A level now we'll look into exactly how those two processes release energy and how much energy is released. So where do we start then? So essentially we'll start by talking about what actually happens in respiration. And here's essentially what happens during the process of respiration. Remember, the process happens inside cells. We associate the process with organelles called mitochondria. What actually happens during the process of respiration? Well, at GCSE, respiration is very simple. It was just glucose plus 6 oxygens made 6 CO2s and 6 H2Os and released energy. That was aerobic respiration. It seemed very simple. At A level, we discovered it's not simple. It's not just simply one reaction step. It's a whole series of reaction steps split into what would appear to be four different stages, as it were. And the first stage of glycolysis, then you have the link reaction, then the Krebs cycle, and then there's something called the electron transport chain. So all these different stages then are involved in the release of energy. Now, where is the energy released from? The energy is released from a glucose molecule. A glucose molecule. So a glucose molecule from AS, you would remember, is a molecule that contains energy. It's an energy molecule. How does the glucose get energy in the first place? Well, you might remember that glucose was made during photosynthesis. It's a molecule that's made during photosynthesis, where you use light energy to construct the glucose molecule from smaller molecules, 6 carbon dioxides and 6 waters, make a glucose molecule, and you get 6 oxygens left over. So you have to remember your GCSE then, your GCSE photosynthesis. And in photosynthesis then, the glucose molecule was made, and it was made using light energy. It needed an input of energy. So energy had to be supplied to plant cells so that they could do photosynthesis and they could make the glucose molecule. So the glucose molecule contains the energy. Essentially, the light energy that the plant absorbed was transferred to the glucose molecule to actually make the glucose molecule. And if you want to think about it this way, the light energy, or the energy that was used in photosynthesis, is now stored in the bonds of the glucose molecule. That's something you probably wouldn't be, your biology teacher probably wouldn't say that to you, but that's essentially a way to look at it, that the glucose molecule has lots of bonds, and those bonds basically contain a lot of energy. If you want the energy out of the glucose molecule, you kind of have to break the glucose molecule down, break its bonds and break it down into bits again, as it were. And by breaking those bonds, you release the energy. So that's how the energy is released in respiration. So essentially, in the 4 stages, in the stages of respiration, and the energy has been released from the glucose molecule. Now, what happens to the energy that's released from the glucose molecule in respiration? Well, essentially, what the cell does is it transfers the energy stored in the glucose molecule to another energy storage molecule. So listen to that carefully. So the cell is transferring the energy that's stored in the glucose molecule. It's releasing that energy and transferring it to another molecule. And the other molecule is called ATP. ATP is a much smaller energy storage molecule. Glucose is a big energy storage molecule. ATP is a smaller energy storage molecule. So the cell is releasing the energy from the glucose molecule and transferring it to the ATP molecule. Now, an interesting question may be asked here is, how much energy is in the glucose molecule? And the answer might be put in a strange sort of way. You might say there's enough energy in the glucose molecule so that when you release that energy, you have enough energy to make 38 ATPs. 38 ATPs. So if you think of it that way, a bit like an exchange rate, all the energy in the glucose molecule is transferred to ATP molecules. And if you can transfer all the energy from the glucose molecule, you get 38 ATPs. Now, when you look at the reactions of glycolysis and the link reaction and the Krebs cycle and so on, you're thinking, it's very hard to see that happening. But essentially, that is what's happening. Essentially, the energy has been taken from the glucose molecule and transferred to ATP molecules. And if you can get all the energy transferred efficiently, you can get 38 ATPs. And that's what happens during aerobic respiration. You get 38 ATPs. And that's the large amount of energy that is mentioned in GCSE that comes from aerobic respiration. If you don't have aerobic respiration, if the cell doesn't have oxygen, it does a kind of an alternative kind of respiration called anaerobic respiration, where not all the energy is released from the glucose molecule. In fact, you only get a very tiny amount of energy released, enough just to make 2 ATPs. So that's a very bad return. Anaerobic respiration gives you a very bad return in terms of the number of ATP molecules that you can make whenever you break down the glucose molecule. Now, you might ask a question, and a question has been asked in exam questions occasionally, is why does the cell do this? Why does the cell transfer its energy from a glucose molecule to these ATP molecules? And you'll see there in the top of the notes at top page 3, that question's asked there, and that question's also present here in the question booklet. And the question's put at the top page 3. It says, why is ATP better than glucose for immediate energy supply? And that was the question that they asked a few years back. And that there's a clue in the question, why is ATP better than glucose? In other words, why does the cell transfer the energy from the glucose to the ATP? And the answer is kind of to put the answer simply, would be to say this, that if you can transfer the energy from the glucose to the ATP is a much handier way of having energy available to the cell. It's handier. You wouldn't say that in the exam, you wouldn't say it's handier. But basically, what you could say is the ATP contains a small amount of energy, which is appropriate to what the cell is using. So the ATP represents a small wee packet of energy, a small amount of energy that's appropriate to what the cell might be using energy for. So for example, a cell might be doing some process that might require 5 ATP molecules. So where the ATP molecule is much handier in the sense that it's a small amount of energy that's appropriate to what the cell, appropriate to the amount of energy that the cell needs at any one time. So rather than the cell having to break down a glucose molecule or extract energy from a glucose molecule every time the cell needs energy, it's already got the energy extracted from the glucose, and the energy is now stored in these much handier, much more accessible, much more efficient little packets of energy called ATP. So the ATP represent small amounts of energy appropriate to the cell's needs. There was a second mark here, and they asked for a second reason. Another good reason is that ATP is more easily transported around the cell. So for example, if you needed energy over at one end of the cell, ATP molecules can be transported over there. If you need ATP somewhere else in the cell, ATPs can move around more efficiently inside the cell. So they're more easily transported, and they've got the right to a small amount of energy appropriate to the cell's needs. And that's a good answer to that question. So that's what the cell's doing. The cell's getting the energy out of the glucose molecule, transferring it to these ATP molecules, which are more handy and more efficient and more useful to the cell and more easy to use by the cell. So that's what the cell is transferring energy from the glucose molecule to the ATP molecules. Now, you have to know the structure of the ATP molecule, and I'm not going to draw it on the board here. It's in the booklet on page 2, and you recognize that it's a molecule that's got 3 phosphates, ATP, adenosine triphosphate, with 3 phosphates. And each of the phosphates are attached to the rest of the molecule by bonds. Now, it turns out those bonds are required. To make those bonds, you have to put in some energy. So to make the ATP, you have to put in some energy to attach the last phosphate to the ADP molecule. So ADP just contains 2 phosphates. If you want to attach the third phosphate, you have to put in an input of energy to attach that third phosphate. And what that means is you've made the ATP molecule. If you want to get that energy back, then you break off the last phosphate off the ATP, and the energy pops out again, and you're left with ADP and P. Okay, so you need to look at that closely. Not something I'm going to talk much more about. It's something really to look at in the notes and understand yourself. It's not too difficult. So I'm going to move fairly quickly on to the more difficult things. So where should we start then? Well, the transfer of energy from the glucose molecule to the ATP molecules takes place in a series of stages then: glycolysis, link reaction, and Krebs cycle, essentially. And those three are happening in sequence, one after the other. So you start with the glucose molecule at the start of glycolysis. It's broken down a bit into two pyruvates, and the pyruvates are converted to acetyl-CoA in the link reaction, and then each of the acetyl-CoAs are then get involved in the Krebs cycle. So you understand that. I'm hoping that you start off with one glucose at the start. You're starting with one glucose at the start here, and then everything ends up doubled up. So you have two link reactions, two Krebs cycles, and so on. And you need the notes to be looking at that and have this sheet in front of you, and you can look at it, and it becomes pretty obvious what's happening. So glycolysis, the link reaction, and Krebs cycle, in that order. In that order, the electron transport chain is another stage or another part of the process. It's not actually a next stage. It doesn't come after the Krebs cycle. The electron transport chain is not in sequence after those three. It's kind of happening all the time. It's happening alongside glycolysis, alongside the link reaction, alongside Krebs cycle. So it doesn't follow after the Krebs cycle, which is often a misunderstanding, students. So here we go. Let's start with the glycolysis. Now, glycolysis, sorry, before we mention glycolysis, just talking about where all these stages happen inside the cell. Well, from AS, we associate respiration with mitochondria. But if you look at it more closely, you discover that glycolysis doesn't happen inside the mitochondria. Glycolysis happens in the cytoplasm of the cell. And in the cytoplasm of the cell, glucose is broken down into two pyruvate molecules. That's glycolysis. And then the two pyruvate molecules, they go in across the membrane of they go in across. So if you have your mitochondria, so that's your mitochondria, and its inner membrane then is folded into these things called cristae, like that. Cristae on the inside. Basically, out here you have glucose being converted to been broken down to two pyruvate molecules, so I'll just call them P, two pyruvate molecules. And then what they do is they go in across the double membrane of the mitochondria, and they go into this region of the mitochondria called the matrix. Okay, so glycolysis happens out in the cytoplasm, and then the two pyruvates go into the matrix, and then in the matrix you have the link reaction, then you have Krebs cycle, and then very importantly, the reactions of the electron transport chain happen on the surface of these cristae. And the cristae are foldings, enfoldings of the inner membrane to increase surface area. Why do you need increased surface area? Well, the way to look at it is each of these cristae sort of represents a surface on which you can position the molecules which are involved in the electron transport chain. So the more surfaces, the more cristae you have, the more surfaces you have for carrying out the electron transport chain. The more surface for electron transport chain, the more the electron transport chain can happen, and the more ATPs, as you'll discover, will get produced inside around the cristae. Okay. So back then to before we move on to glycolysis, how do you know that a cell is doing a lot of respiration? What's the evidence that a cell might be doing more respiration than other cells? The answer is the number of mitochondria present in the cell. So if you find a cell with a lot of mitochondria, that suggests there's a lot of respiration taking place in that cell. This was a 4-marker question in a recent essay. The second thing you would notice is that the mitochondria might be very big. So if you've got very big mitochondria and a lot of them, then that's 2 indications there's a lot of respiration taking place. The third thing is if the mitochondria have a lot of cristae present, if they have a lot of foldings, on the inner membrane, then it's an indication, further indication that there's a lot of respiration taking place. So there's 3 indications. Lots of mitochondria, if they're big mitochondria, and lots of cristae foldings. All those are indications that the cell's doing a lot of respiration. Okay, let's move on then to the first stage in respiration, which is glycolysis. And glycolysis, essentially, in glycolysis, glucose is broken down through a series of steps, 4 or 5 steps, into 2 molecules of pyruvate. Notice the number of carbons. Glucose has 6 carbons, and then eventually it's broken down into 2 pyruvates, and each of the 2 pyruvates just have 3 carbons. All this is in the notes here and done in tremendous detail, not in tremendous detail, but in the amount of detail that's required to get it across very clearly to you. So you definitely need the notes to be able to follow what I'm doing here. So you've got your 2 pyruvate molecules. Now, question students will ask me is, how much of glycolysis do you need to learn? Do you need to learn all the steps? And an answer to that question is, no, you don't need to learn all the steps. By all means, learn them if you want. But you need to know the essentials. You need to know that it starts with glucose. And you need to know that it finishes with 2 pyruvates. The glucose has 6 carbons and the pyruvates have 3 carbons. In between, you need to know 2 things, a couple of things. You need to know that in the very first step, there's an input of ATP. In other words, ATP molecules are required. And in the very first step, you have to put in 2 ATP molecules. So this is a bad start to glycolysis. You're having to supply, the cell's having to supply 2 ATPs to get the process going, as it were. And then in the very last step, however, you get 4 ATPs made. So you've used up 2 in the first step, but in the last step, you make 2 ATPs in each of the 2 steps, because you're getting 2 pyruvates, and that's a total of 4 ATPs. So the overall output or the production of ATPs for glycolysis is 2 used up at the start, 4 made at the end, overall plus 2. So you get basically, glycolysis gives you 2 ATPs. And just when we're at that, just when we're saying that, if you remember, anaerobic respiration just gives you 2 ATPs. So maybe anaerobic respiration is just glycolysis. Essentially, that's what it is. In anaerobic respiration, glycolysis is the only stage that actually happens. The other stages don't happen. So that's why anaerobic respiration gives you two ATPs. The 2 ATPs is from glycolysis. Yeah? Okay. We'll come back to anaerobic respiration. Maybe not even in this video, so we mightn't get that far. So, glycolysis, what sort of question do you get then? Well, as I say, you don't have to learn the stages in glycolysis. The best example of a question there is question 6 or question 7. Let's see if I can find it there. Yeah, I'm looking at question 8 in my question booklet, and I use this question always to students to say this is a good glycolysis question, give you an idea what's required. And you'll see in this glycolysis question, it tells you that in the first step 2 ATPs are used up. And then it asks you, then the question is this, part A asks you, indicate on this, it shows the sequence of steps in glycolysis, finishing with 2 pyruvates. And it says, indicate on the diagram where ATP is made. And all you do is you put an arrow in the last step, saying ATP is made in the last step, because there's 4 ATPs made in the last step. But the other thing it asks is, indicate where NADH is made. Now, NADH is can be called various things, and it doesn't really matter what you call. You can call it NADH, or you can call it reduced NAD, okay, or you can even say NADH2. I prefer to use reduced NAD. Reduced NAD. Reduced means that the NAD is attached to hydrogens. NAD has got hydrogens attached, and in that state it's called reduced NAD. If it had no hydrogens attached, it's just NAD, or you could call it oxidized NAD. Okay, so that's then where's reduced NAD made in this step, and that's the second last step. You have to indicate with an arrow. I've never seen a question where you actually had to fill in any of the stages of glycolysis. Nothing like that. I've never ever seen that, and I've been marking exams now for 20 years. So they tend not to ask. They're not leave one out and ask you what it is. They tend not to do that. That's the sort of thing that ask you about glycolysis. What you'll probably discover is they don't ask about glycolysis much at all because it's too easy. Usually if you get an exam question, it's about anaerobic respiration or something else much more definite. So don't spend too much time learning glycolysis. Essentially, that's the end of glycolysis. What have we said about glycolysis? Overall, there's just 2 ATPs made, and glucose has been broken down into 2 pyruvate molecules. Okay, and this happens in the cytoplasm. What then happens is the 2 pyruvate molecules go in across the membrane of the mitochondria into the matrix. And the next stage happens here. And what is the next stage? The next stage is called the link reaction. And the link reaction is very, very straightforward. You may have complicated notes from school, and it may look complicated in other places, but essentially all you're saying in the link reaction is that your pyruvate, PYRUVATE, which has got 3 carbons, is converted into a molecule called acetyl-CoA, ACETYL-CoA, acetyl coenzyme A, and it's got 2 carbons. And that happens to each of the pyruvate molecules. Remember, you have 2 pyruvate molecules, each of them is converted into acetyl-CoA. You'll notice that you go from 3 carbons to 2 carbons. And that's an indication then is that there's a carbon lost, and the carbon lost is CO2. So there's a CO2 molecule released in this step. So how many molecules are released in the link reaction? 2 CO2s, because the step happens twice. CO2, CO2. Okay, and the other step as well. The other thing that happens is you also get a reduced NAD. What happens is NAD, hydrogens get released in this step as well, and the hydrogen joins up with NAD, so you get NADH, or reduced NAD. So those are the 2 things that are reduced here. You get carbon dioxide released, and you also get hydrogens released, and the hydrogens are picked up by this thing called NAD. And NAD is simply just a carrier molecule. It's just a molecule that picks up hydrogens and carries them away somewhere. Where does it carry the hydrogens away to? It takes them to the electron transport chain. NAD is just like a delivery vehicle, a delivery thing that picks up hydrogens wherever the hydrogens are released and carries them to electron transport chain. So there's NAD picking up hydrogen there and taking away the electron transport chain. So, by the way, sometimes they'll use terms, and this is not a term you need to know, but the release of carbon dioxide is called decarboxylation. Decarboxylation's on the notes there. And the release of hydrogen is called dehydrogenation. Okay. Now, you also maybe come across the idea that these steps involve enzymes. So you have enzymes that remove carbon dioxide that allow this step to happen, and they're often called decarboxylases. And enzymes that hydrolyze the removal of hydrogens, and they're called dehydrogenases. So we'll mention those again later on. But all of this is mentioned in the notes there. Okay. So that's glycolysis, and also the link reaction then. Those are the first 2 stages. So if I was to ask you how many CO2s have been released now since we started with glucose, we've had 2 CO2s released, CO2 here and CO2 in the other step, the other pyruvate 2 acetyl-CoA step. So 2 CO2 released. So on we go then, moving fairly fast. Just checking, I think we're running about 40 minutes so far. Moving on to the next step, which is the Krebs cycle. Okay, and the Krebs cycle is pretty straightforward. Essentially, the acetyl-CoA feeds into the Krebs cycle. And if I can just do a quick diagram of the Krebs cycle. So remember what you have so far. You've now got 2 acetyl-CoA molecules produced. 2 acetyl-CoA molecules produced. Now, what happens next is if we have a box here, we'll put acetyl-CoA here. Here's the box, acetyl-CoA, ACETYL, CoA. And if you remember, it has 2 carbons. 2 carbons. Yeah, essentially what happens, and this is the way I approached this just to get you a diagram in the notes. So this is a good approach for you to use as well. If you put a box here for acetyl-CoA, you can actually draw out the details of the Krebs cycle. So if you put maybe a box there, and a box there, and then put a box down the bottom. So if you just do this on a sheet of paper, you'll find this is a useful approach. And a box there, maybe not exactly in the right place, another box there, and so on. And essentially what you do is you start over here and you go 4 carbon acid. A 4 carbon acid. Okay, you don't need to know its name, it's just a 4 carbon acid. Some schools will tell you to learn the names of these acids. I've never seen them asked. I've never seen a situation where students are asked, what is the name, what is that acid, or what's this acid? So it's a 4-carbon acid, and here's the first step then. The first step involves your acetyl-CoA, which comes from the link reaction, combining with this 4-carbon acid. So the way you do that is you sort of put a line across there, and you bring down the acetyl-CoA like that. So the acetyl-CoA combines with this 4-carbon acid, and the two of them together make, what do they make? They make a 6-carbon acid. 6-carbon acid. So you've got 4 carbon and 2 carbons make 6-carbon acid. Okay? And then all you need to do, and this is quite easy to learn then, if that's 6-carbon acid, you make this a 5-carbon acid. Okay? And then you just make this a 4-carbon acid. And you're down to 4 carbons, you can't go any lower, so this is another 4-carbon acid. So look at how easy that was. Make yourself all those boxes, and then watch what happens. Take the acetyl-CoA, which is 2 carbons, join it with a 4-carbon, you get a 6-carbon acid. And then the 6-carbon acid is converted to a 5-carbon acid. The 5 is converted to a 4. The 4 stays as a 4, and it stays as a 4. Now, notice the number of carbons is very important here. If this is a 6-carbon acid and this is a 5-carbon acid, guess what? You get a CO2 released here. CO2, yes, that makes sense. You're going from 6 carbons to 5 carbons. So there has to be a CO2 released. And look down at this stage, from 5 carbon to 4 carbons. Guess what? Another CO2 released. Yep. So if we just stop at this stage and I say to you now, how many CO2s have been released since the start of respiration? And you say to yourself, okay, when was CO2 released in respiration? Oh, the link reaction, yes. There was CO2 released in each branch of the link reaction. So that was 2 CO2s, okay. And then there's 2 CO2s released here. But this happens twice, of course. I mean, you get 2 Krebs cycles, don't you? Because you have two acetyl-CoAs. So this happens twice. So 2 CO2s here by 2 gives you 4. And then the 2 from the 2 branches of the link reaction gives you a total of 6 CO2s released from released so far. 6 CO2s released. And this is what you expect. Well, I'm saying, how can you expect there's going to be 6 CO2s released? Well, if you go back to GCSE and remember the equation for respiration, it was glucose, which was C6H12O6, plus 6 O2s gives you 6 CO2s and 6 H2Os. So here's where the 6 CO2s come from. So the 6 CO2s, 2 of them come from the link reaction, and 4 of them come from the Krebs cycle. Yeah, are you okay with that? Are you okay with the idea that the Krebs cycle happens twice, and the link reaction happens twice? So each step of the link reaction gives a CO2. That's 2 CO2s, and each Krebs cycle, each turn in the Krebs cycle gives you 2 CO2s. That's 4 and 2, 6, so a total of 6 CO2s. And if you look back through the past paper questions, I think that actual past paper question might be in the booklet here, or I may have removed it, but it's a fairly recent, I think it's been 2018, where they asked you to account for the 6 CO2s that are released whenever each glucose molecule is broken down in respiration, and you had to add them all up and show that there were 6. It's quite an easy question. It's a very easy question. Okay. Now, what the Krebs cycle? Now, would I advise you to know that there? Yeah, I think that's a good thing to know. To know those, know the sequence there. You don't need to know the names of the acids. I've been marking this for a long time, and I've never once seen them asking you about the names of the acids. It's important to know the number of carbons the acids have. And look at the pattern. 2 plus 4 gives 6. 6 goes to 5 with CO2 released. 5 goes to 4, another CO2 released. And then you just stay 4, so there's no more CO2s released then. Now, the other thing that you need to know then about the Krebs cycle is that there are other things released then. And here's what the other things that are released. You get released here you get some hydrogens. So in this step you get some hydrogens there, and you get some hydrogens released here. Okay, and over here you get some hydrogens released, and over here you get some hydrogen released. So notice there are 1, 2, 3, 4 points where you get hydrogen released. Okay, now you're thinking, do I have to remember those points where the hydrogen released? And the answer is no. You don't have to remember exactly where they're released. All you have to know, what I would say you have to know is, know that there's 4 occasions, 1, 2, 3, 4 occasions where you get hydrogen released. 4 occasions. Okay, and what happens to those hydrogens? Well, those hydrogens are picked up on each occasion by NAD. So this hydrogen is picked up by NAD, and you get NADH. There we are, that hydrogen. And this hydrogen is picked up here, so you also get NADH. There we are. And this hydrogen is picked up by NAD, so you get NADH. Or as I say, I prefer to call it reduced NAD. But this is the pinch here. This particular hydrogen is not picked up by NAD. It's picked up by another carrier molecule called FAD. FAD. Now you don't have to know the difference between NAD and FAD. Just think of it as a different carrier molecule. It's just like NAD, it's just a different carrier molecule. So notice that again. Notice we've already said you get 2 places where CO2 is released. Yeah, we forgot about those. Now let those go. And we've got 1, 2, 3, 4 places where hydrogen's released. And what happens to those hydrogens? On 3 of the occasions, the hydrogen's picked up by a carrier called NAD. In other words, you get what's called reduced NAD. NAD with the hydrogen attached, then it's reduced NAD. And on the other occasion, it's picked up by a different molecule called FAD. So 4 occasions where hydrogens are picked up. And that's nearly it. And that's nearly everything we need to say, except one last thing, and I'm looking for a slightly different color here. In one of the steps, you get an ATP molecule raised. And it's that step there. And do you have to remember that it's that step? Yeah, possibly. But not necessarily. You would have to know that there is, if they showed you a diagram where they said CO2 been released, NADH been released, and they said what's molecule X, and they just put an arrow and you would say, oh, molecule X, that's a little ATP molecule. So an ATP molecule. So what all happens? This is the Krebs cycle then. What happens in it? Here's the way I approach it. I say, and this is all in the notes, very clear. The Krebs cycle just involves a series of steps involving acids. In 2 of the steps, carbon dioxide is released, so that's 2 CO2s. In 4 of the steps, hydrogen is released. 3 of those hydrogens are picked up by NAD, and the other hydrogens picked up by FAD. Oh, and one last thing, in 1 of the steps, an ATP molecule is released. So if I said to you, how many ATPs are released in total in the Krebs cycle? The answer is 2. Why is it 2? Because the Krebs cycle happens twice, doesn't it? For every glucose molecule, 2 ring reactions, 2 Krebs cycles, so you get a little ATP. So in total you get 2 ATPs released in the Krebs cycle. So if you count now from the start, how many ATPs have you got so far? Remember how many ATPs you wanted from each glucose molecule. If everything was going good and you had oxygen available to the cell, you expected 38 ATPs. You're expecting 38. It's not going so well so far, is it? In glycolysis, you just ended up with 2 ATPs. The link reaction, oh, there was no ATPs at all in the link reaction. So you had 2 in glycolysis, none from the link reaction, and now you have 1 from each Krebs cycle. So in total, so far, you just have 4 ATPs. That's not very good going so far. You expected 38. We've only got 4. We need another 34 out of somewhere quickly. And the question is, where's the rest of the ATPs going to come from? And here's the simple answer. You see all these NADs and FAD and NADs that were in the link reaction and the NADs that were in the glycolysis reaction. All those NADs are going to bring their hydrogens to where? They're going to bring their hydrogens to the electron transport chain. The electron transport chain. And in the electron transport chain, guess what? You get the other 34 ATPs that are missing so far. You get the rest of the 34 ATPs. And it's the electron transport chain that requires the presence of oxygen. The electron transport chain needs oxygen. So you're only going to get those other 34 ATPs if the cell has oxygen available. If the cell doesn't have oxygen available, you won't get those 34 ATPs in the electron transport chain. And it turns out you won't get these ATPs from Krebs cycle either, and so you're just left with only getting 2 ATPs from glycolysis, and I'll explain that later to you. But essentially you're expecting to get the other 34 ATPs from the electron transport chain when these hydrogens are brought to the electron transport chain. Okay, sorry for going so fast, but there's a lot of stuff to talk about. So there's the Krebs. So your approach to the Krebs cycle should be, I would say to you, just get some sheet of paper, draw these boxes. How many boxes? One at the top, two, and one at the bottom. Put the acids, put the silicate, and then try and join them up yourself. And you'll discover it's pretty straightforward. Okay, good. Now, do you get ever asked to draw the whole of the Krebs cycle? No, not at all. But if you have the questions in front of you here, I'm going to point at questions where you're asked to say, it's telling us here in the Krebs cycle, you get NAD, FAD, ATP, and then it asks you what's molecule X, what's the other molecule that you get in the Krebs cycle, and the answer is your little ATP, and that's it. and then it asks you just about what the pathway is called. By the way, pathway is just another word for stage. Like glycolysis is a pathway, and then link reaction is a pathway, and Krebs cycles a pathway. Sometimes the older questions have odd ways of asking things. And in this other question that it's just showing pictures of the acids, and it's asking you what is molecule X that's released during the electron transport chain, and so on and so forth. And there you are. That's the sort of question that you get asked about the Krebs cycle. So in general, you don't get asked to reproduce glycolysis or the link reaction or the Krebs cycle, but you have to understand. You have to know where certain molecules are released, like carbon dioxides and hydrogens, and who picks up those hydrogens, and where do they go to? So they go to the hydrogens, go to the electron transport chain. What happens to the carbon dioxides? Well, the carbon dioxides are released from the mitochondria into the cell, and then they're released from the cell to be lost from the body. So, for example, if your cells are doing respiration, they're making carbon dioxide. That carbon dioxide diffuses out of the cells into the bloodstream, and the carbon dioxide then is carried to your lungs. And when you breathe out, the carbon dioxide comes out. That's where the carbon dioxide that we breathe out comes from. It comes from the link reaction and Krebs cycle. The carbon dioxide is just there. Now, so as I say, you absolutely need these notes in front of you, and it's easy to get the notes. You just get in touch with me and I'll send you a package of stuff, and then you can sit and watch this video if you want, and it'll make a lot more sense to you. Also, if you want to get a package of notes, you can Skype with me, sure, and we can talk through the questions and talk through the answers and all that sort of thing. But okay. So I think that's probably as much as I want to do so far. I'm going to leave the rest of the respiration topic then for another video, because I think I'm just looking at the running time here. We'll run about 45 minutes, and that's probably as much as I can do without needing a drink of water here. So I'm going to stop at this stage. But essentially, to say to you, the rest of the respiration topic is pretty okay. It's pretty straightforward. You just have to know how to learn it and know what approach to take, and to know what bits of it are, what bits you need to learn off by heart, and which is not very much of it, and what bits are not too important. And that's where I come in. Essentially, I'm trying to just include in the notes the key bit, the bits that you need exactly set out, and also when we go through the questions, the key thing is the questions. By going through the questions, you see exactly what you get asked over the years, and then you develop an understanding that you only tend to get asked certain things, and there's really a limited amount of stuff that you can get asked. So it's useful. It's definitely useful to know these things. I also produce here a set of what I call short questions. There's about 50 questions on respiration, but they're not by any means short. Some of them are short. Some of them ask you simple facts like, here's a fact: name the nitrogenous base present in ATP. The base present in ATP. Bases in ATP. Surely bases are to do with DNA. Well, no. ATP contains the same base as you get in DNA. The base adenine. That's where an ATP adenine. The first bit is basically adenosine, adenine. So that's the sort of question you get in here. I've asked most of the exam questions that have come up over the years. I've asked them in sort of short form in this booklet here, and you have all the answers. All the answers are here, so you can go through the booklet and check yourself out. So I've asked here, for example, how many CO2 molecules are made during glycolysis, and you have to write, and the answer is 0. How many are made during the link reaction? And the answer is 2. And how many are made during oxidative phosphorylation? Oh, we haven't done oxidative phosphorylation yet, but basically that's the electron transport chain, and the answer is 0. Okay, so these questions are good testing questions, which are really good for your understanding and also good for your confidence. If you can go through these questions at the end of this topic and answer them all, there's 57 questions, and none of them are outrageous. Most of them are taken from the notes. Most of them are taken from past paper questions. In some cases, they're ones that I have dreamt up, sort of suggest type questions. And I've been marking A-level exams now for 20 years, so I kind of have a feel for the sorts of questions that they might ask that they haven't asked before. So going through these questions in it is definitely a good thing to do. Okay, I'm going to stop there. There should be a later video on the rest of respiration, which should take in the electron transport chain, which is very important, and anaerobic respiration, and alternative respiratory substrates, which is looking at how we can also use proteins and fats for respiration, and then the experimental stuff at the end, the respirometer and that, and I'll be looking at that as well. So, okay then, thank you very much, and if you need notes and stuff, you can get in touch with me there. Okay, bye.